Welcome to Pathogenic Microbiology 2. I am so excited. This is my favorite subject, and we are going to learn so much. And I'm so excited to share my knowledge and my passion and love for microbiology with you. This chapter 1, I shouldn't tell you this, but there will not be any test questions that come from this lecture. This lecture will serve as the foundation of everything we're going to learn in this semester of pathogenic microbiology. We're going to learn about bacteria. We're going to learn genus and species, a lot of names, a lot of terms. So this first set of lectures from chapter 1 is just to set the foundation, teach you some terms, and give you a foundation so you can understand the importance of what medical laboratory scientists and technologists and technicians do in the laboratory. So I started out with a joke about the 5-second rule. There's a potato chip that's dropped on the floor, and the bacteria are saying, "Don't touch that chip yet." 1, 2, 3, 4, too late. So there is no such thing as the 5-second rule. All right, to begin with, our book. This textbook includes case studies throughout the book, and these case studies will help develop critical thinking skills, which are crucial in clinical microbiology. I strongly suggest that you read the case studies yourself, even if we don't cover these in class. They will help you a lot. So here's just an example, and I'm not going to read it. We are going to get into that. It's going to be so exciting. All right. Here are a list of key terms. These will be in your book, and I suggest strongly that you read your book before the lectures or after to help clear up points, and it will definitely help. Okay. I love this picture because this is a typical microbiology workbench, work area where a medical laboratory technician or scientist will work. You can see she has a patient culture right here. This is called a split plate. It's an agar petri dish, and it has half of the agar is probably a blood agar, sheep blood agar that grows just about all kind of bacteria. And this is most likely a MacConkey agar that grows a little bit different kind of bacteria. We're going to get into that later. So you can see the streaking pattern right here. That's called a colony count. We're going to get into all that. It's so exciting. But look, here is all the agar that you get to work with. All the different media, the different colors represent a different kind, and you cannot tell, but this is actually a different size. This is a really large plate. That you could do like a water colony count on. These would be broth tubes here, different kind of broth media for bacteria to grow in. This is another kind of broth. She has her, we call this a toolbox, a few quick latex reagents right here to just quickly do some biochemical testing. Everything's set up there that she needs. And look, a Sharpie. Of course, we all need a Sharpie. Buy yourself some Sharpies before you start clinicals. So here comes the really important part. What is the role of the clinical microbiologist? What do we do? Well, first we isolate bacteria causing disease. Then we identify the bacteria causing a disease, giving it a name to genus and species. Then we analyze the bacteria, which means we figure out which antibiotics can be used for treatment. In order for us to fulfill these goals, we have to have a solid foundation knowledge of microbial structure and physiology. So structure would be the cell wall, what it's made out of, and physiology is the kind of biochemical pathways that it uses to metabolize things like glucose or other carbohydrates that it eats. So this allows us to, it causes us, these are what we're trying to do. So how do we do that? We culture organisms from specimens that we get. Then we classify and identify organisms, and I'm going to show you how to do that. That's what we're going to be doing mainly in this course. And then that allows us to predict and interpret susceptibilities to specific antibiotics. So in a nutshell, that's what we are doing. We are classifying the bacteria, identifying it, and then we're telling the doctor, hey, this is the bacteria that's causing your patient's infection, and here's the antibiotic that will kill it or not. And really the most important thing that we're looking for is resistance, that bacterial resistance to antibiotics that in the past have been used perfectly fine. So we're looking for emerging drug resistance. That's what we got to keep a lookout for. So in order for us to know all this, we have to know microbial structure and physiology, and that is going to be the bulk of what is you're learning in microbiology. So exciting. It's my favorite. Okay. So how are we going to grow these things in the lab? The first step to successfully culturing organisms from patient specimens is to understand the growth requirements of many bacteria and select the correct medium, temperature, and atmosphere. Do they need oxygen or not? And this is going to be our primary culture conditions. And knowing these, the agar, the temperature, the atmosphere that the bacteria like, is going to increase the likelihood that we can grow and isolate that pathogen. Bacteria also grow at different rates, so that's another clue that's going to help us. Some bacteria, like E. coli, can double or replicate once every 20 minutes. So you start out with 1 E. coli, and in 20 minutes you have 2, and in 20 more minutes you have 4, and in 20 more minutes you have 8. You get it. So it only takes hours before the bacteria can be seen on the medium. Other bacteria, like Mycobacterium tuberculosis, take a whole 24 hours to replicate just once. So it takes weeks before there's enough Mycobacterium on the agar or medium so that we can actually see the different colonies. Bacteria also require different atmospheric conditions, meaning oxygen content or lack thereof. So we're going to talk about how you can incubate in increased carbon dioxide and in zero oxygen and different degrees or percentages of carbon dioxide. It's going to be very interesting. So first we're going to start the overview of the microbial world with bacteria. They are prokaryotes, which means their cellular walls are different from ours. Parasites and fungi are both eukaryotes, but fungi are heterotrophic eukaryotes, which mean that they get their nutrients through absorption. Fungi are composed of yeast and molds. Yeast are unicellular fungi that reproduce asexually through a process called budding. They do not have mycelia or hyphae. Hyphae are filaments that can give fungi a fluffy appearance, and mycelia are flat mats of interwoven hyphae. Some fungi can be a yeast at one temperature, for example, body temperature, and the filamentous mold form at room temperature. So when a fungi can exist in both morphologies, it is said to be dimorphic. You guys knew that already. Viruses are obligate intracellular parasites that can't be seen with the regular light microscope. You need an electron microscope to see a virus. Viruses have some type of nucleic acid, either DNA or RNA, but never both. And they are usually cell-specific. For example, HIV virus attacks T helper cells and only T helper cells. So I think that's enough information for the first part. We are going to pause right here at the end of Lecture 1, Chapter 1, Part 1.